Let's get right into it. The top 10 animals with superpowers. This four-winged killer flies in at number 10. In the realm of Earth's ancient and diverse creatures, the dragonfly stands out as a paragon of evolutionary success, embodying traits that seem almost otherworldly in their precision and efficiency. Belonging to the order Anisoptera, these insects have navigated the challenges of Earth's changing environments for over 300 million years, evolving capabilities that not only ensure their survival, but also position them as apex aerial predators in their ecosystems. Dragonflies are renowned for their extraordinary flight capabilities. Unlike most other insects, dragonflies possess two sets of wings that they can operate independently, allowing for precise maneuvering in mid-air. This wing structure enables them to perform a variety of aerobatic feats, including hovering, darting, and even flying backwards. Each wing can pivot at its base, enabling sharp turns and sudden stops that confound both prey and predators. The dragonfly's flight muscles are uniquely structured, attaching directly to the wings rather than to the body, which allows for greater force and control in wing movement. The speed and agility of these creatures are further enhanced by their wing beat frequency, which can vary between 30 to 40 beats per second. This rapid wing flapping generates the power needed for quick bursts of speed, with dragonflies reaching velocities of up to 35 kilometers per hour. Additionally, their flight endurance is remarkable. Certain species migrate across oceans, covering thousands of kilometers. The globe skimmer dragonfly, for example, migrates from India to Africa, a journey spanning over 18,000 kilometers, relying on high-altitude winds to sustain its long-distance flight. Dragonfly vision is another aspect where they exhibit exceptional capabilities. Their compound eyes, among the most elaborate in the insect world, consist of up to 30,000 individual ommatidia photoreceptor units. These eyes provide a nearly 360-degree field of vision, which is critical for detecting movements of other flying insects from various angles. The large size of the eyes relative to the head and the high resolution of their vision allow dragonflies to discern colors, polarized light, and ultraviolet light, further aiding their hunting prowess. Research indicates that dragonflies can identify and track moving objects separated by just 0.04 degrees, which means they can spot a mosquito or a fly from meters away, despite the small size of these targets. Dragonflies begin their life cycle in water, where they spend months or even years in a larval state known as a nymph. These nymphs are equally formidable predators, equipped with a unique extendable lower lip, labium, that they can thrust forward to snatch prey such as tadpoles, fish, and other aquatic larvae. The transition from nymph to adult, an event marked by the dramatic process of metamorphosis, is a critical phase during which the nymph climbs out of the water, sheds its exoskeleton, and emerges as a winged adult. This transformation not only symbolizes a change in habitat, but also a new phase in their predatory lifestyle. Deep within the ocean's abyss at number 9, where light fades and the mysteries of the sea dwell, the sea cucumber exhibits capabilities that seem to border on the supernatural. This creature, scientifically classified as Holothuroidea, possesses a set of survival mechanisms so advanced that they capture the awe of those who study the depths of our oceans. Among the most extraordinary of these capabilities is the sea cucumber's defense mechanism of evisceration, where it expels its internal organs to thwart predators. This might sound like an act of desperation, but in the domain of the sea cucumber, it is a calculated strategy for survival. When threatened, certain species of sea cucumbers can expel a tangled mass of tubules, their Cuverian tubules, from their anus. These sticky and sometimes toxic threads can entangle and deter predators such as fish and crabs, allowing the sea cucumber to escape imminent danger. What is truly miraculous about this process is not just the act of evisceration itself, but the sea cucumber's ability to regenerate what has been lost. Depending on the species and environmental conditions, sea cucumbers can regenerate their lost organs in a span ranging from a few weeks to several months. For example, researchers have documented that some species can regenerate their digestive digestive tracts fully within 14 to 17 weeks, a feat that involves complex biological processes involving stem cells and growth factors. These cells multiply and differentiate to form not only the structures that were lost, but also the intricate networks of nerves and muscles associated with them. The mechanism behind this rapid regeneration is as fascinating as the process itself. Sea cucumbers possess a unique collagen framework within their bodies that provides both structural support and incredible flexibility. This mutable collagenous tissue can change its mechanical properties from stiff to soft under neural control or in response to external stress. Such a transformation allows the sea cucumber not only to liquefy its body to escape tight grips, but also to rebuild its structural integrity during the regeneration phase. Moreover, the internal environment of the sea cucumber supports this regeneration through a highly efficient biochemical pathway that synthesizes collagen and other essential proteins at remarkable speeds. The presence of growth factors and cytokines in their tissues further accelerates healing and regeneration, ensuring that the sea cucumber returns to its original state in a relatively short period. From an ecological perspective, the sea cucumber plays a crucial role beyond its sensational survival tactics. These creatures are integral to the nutrient cycle of marine ecosystems. As they feed by ingesting sediment, they break down detritus and other organic matter, recycling it back into the ecosystem. This process not only cleans the ocean floor, but also facilitates the proliferation of microorganisms and the distribution of nutrients across the seabed. The sea cucumber's repertoire of abilities, from its self-defense to its regeneration and ecological contributions, paints a picture of a creature that, though lacking the flamboyance of more well-known marine animals, possesses a suite of powers that are critical to the survival and health of oceanic ecosystems. These capabilities, lying at the intersection of bizarre and scientifically fascinating, underline the sea cucumber's role as a marvel of evolutionary innovation and an enduring subject of scientific study. In the shadowed recesses of New Zealand's limestone caves, a spectacle akin to the starlit sky unfolds, but this celestial mimicry is not without its deadly intent. Here, the glowworm, or Arachnocampa luminosa, stages a light show that is both mesmerizing and lethal, employing bioluminescence to execute a predatory strategy as brilliant as it is brutal. Like a holographic Charizard coming in at number 8, the glowworm is not a worm, but the larval form of a fungus gnat, and it is in this stage that the creature reveals its most remarkable trait. Dangling from cave ceilings, these larvae emit a soft sapphire light from their abdomen, a process driven by a biochemical reaction within their bodies. This light is the result of luciferin, a molecule that, when oxidized in the presence of the enzyme luciferase, produces a radiant blue-green glow. This luminescence serves as a beacon to attract flying insects, which are lured into the deceptive safety of the glowworm's habitat. However, the glowworm's cave is no haven, but a deadly trap. Suspended from the roof, each larva spins a series of silken threads up to 70 centimeters long. These threads are coated with sticky droplets that reflect the larva's bioluminescent glow, enhancing the trap's allure through scattering light. The physics of this light attraction is not merely passive. The glowworm can modulate the intensity and pulsation of its glow to maximize its appeal to different insects, which perceive light in various spectrums. This ability to adjust is a sophisticated evolutionary adaptation that targets a broad range of prey. The effectiveness of this strategy is astonishing. Research suggests that the brighter the larva's glow, the greater its success in attracting prey. This luminous lure is not just about intensity, but also about the economy of energy. The biochemical process of bioluminescence in glowworms is remarkably efficient, converting nearly all the energy used into light without losing much as heat. This efficiency is crucial in the nutrient-poor environments of the caves where they reside. Once an insect makes contact with the silken threads, it becomes ensnared, and the glowworm begins its feast. The larva uses its mouthparts to haul in the caught prey, subsequently consuming it, sometimes over several hours, depending on the size of the catch. This feeding process is vital, as the energy derived from their prey supports them through their larval stage, which can last up to several months, and into their pupal stage, where they metamorphose into adult gnats. Interestingly, the glowworm's life as an adult is starkly different and devoid of the luminous glory of its youth. Adults do not feed and live only a few days, with their sole purpose being reproduction. This dramatic shift highlights the unique adaptation of the larval stage, where all the creature's predatory capabilities abilities are maximized. Moreover, the glowworm's bioluminescence plays a crucial role beyond predation. It is also a deterrent against potential predators of the glowworms themselves, who may be repelled or confused by the light. Additionally, in dense populations, their collective glow can influence the microclimate of their environment, possibly deterring the growth of mold and bacteria on cave surfaces. This creature can reverse time itself at number 7. Amidst the marvels of the marine world, the immortal jellyfish, Turritopsis dohrnii, showcases a seemingly supernatural ability that captures the essence of eternal youth. This diminutive creature, often no larger than 4.5 millimeters in diameter, possesses the extraordinary capability to revert from its adult form back to its juvenile polyp state, a process that potentially grants it indefinite life, barring external fatal factors. The life cycle of Turritopsis dohrnii is a fascinating journey. Typically, a jellyfish starts as a tiny larva known as a planula, which after settling onto a suitable surface, develops into a colony of polyps. These polyps then bud into free-swimming medusae or adult jellyfish. However, the immortal jellyfish diverges from this usual path in response to stress or physical decline, invoking its unique ability to transform its cells. This transformation involves a rare biological process called transdifferentiation, where its cells can change from one type to another, allowing the adult medusa to revert to the polyp state. This phenomenon is not just a mere survival tactic, but a reversal of the aging process itself. During transdifferentiation, specific cells of the outer layer of the jellyfish, known as the epidermis, and cells from the lining of the digestive canal can morph into various other cell types. For instance, muscle cells can become nerve cells or even revert to a more primitive, undifferentiated state, essentially rebooting the life cycle of the organism. Remarkably, this cycle of rejuvenation can repeat indefinitely, rendering the jellyfish biologically immortal. This capability of transdifferentiation is highly unusual and not observed in any other animals known to science. Turritopsis dohrnii's ability to practically bypass death and reset its life is a sophisticated adaptation, evolved perhaps as a response to the unpredictable and often harsh conditions of the marine environment. In habitats where predation, competition, and the chance of death are high, the ability to revert to a juvenile state allows another chance at life, providing an exceptional survival advantage. Scientific studies have shown that the environmental triggers leading to this rejuvenation process could include physical damage, starvation, temperature changes, or chemical changes in the surrounding water. Each of these factors can initiate a complex genetic and cellular response that leads to the jellyfish's transformation. From an ecological and evolutionary perspective, Turritopsis dohrnii's immortality is of significant interest. This jellyfish's distribution has expanded across various oceans, including the Mediterranean, the Atlantic coast of Panama, Spain, and Japan. This wide dispersion is attributed not only to its survival and reproductive strategies, but also to its stowaway travels in the ballast waters of ships. The study of Turritopsis dohrnii extends beyond biological curiosity. It holds potential implications for human medicine, particularly in the fields of aging and regenerative medicine. Understanding the molecular and cellular mechanisms underlying its transdifferentiation could illuminate new pathways for tissue regeneration and longevity research, potentially leading to advancements in treating age-related diseases and injuries. Like Go the Sniper in Halo 2, this sharpshooter comes in at number 6. In the intricate web of nature's marvels, the Bombardier Beetle, Pheropsophus verticalis, exemplifies an evolutionary masterpiece of chemical warfare. This small beetle, typically just a few millimeters long, possesses an astounding defensive mechanism that allows it to confront threats in a manner unparalleled in the animal kingdom. This beetle is equipped with a highly specialized internal structure that houses 2 separate chemical reservoirs within its abdomen. These reservoirs contain hydroquinone and hydrogen peroxide, 2 relatively harmless substances on their own. However, when the beetle feels threatened, it triggers a defense response that is both violent and precise. The secret to the Bombardier Beetle's defense lies in the reaction chamber that is anatomically designed to mix these chemicals. The enzymatic breakdown of hydrogen peroxide by catalyses and peroxidases generates free oxygen and significant amounts of heat. This rapid decomposition elevates the temperature of the mixture to nearly 100 degrees Celsius, effectively boiling the concoction inside the beetle's specialized reaction chambers. What follows is a controlled, explosive reaction reaction. The heat causes the hydroquinone to undergo oxidation reactions, transforming into p-benzoquinone, a highly irritating compound. This reaction also generates gas, increasing the internal pressure exponentially. At just the right moment, the beetle releases this pressurized hot chemicals spray through a nozzle directed at its predator. The nozzle can articulate, spraying in a wide range of directions with an accuracy that allows the beetle to target threats directly, even in mid-flight. The chemistry involved in this defensive spray is finely tuned. The spray is emitted in a series of rapid, pulsating bursts, which scientists believe is a mechanism designed to optimize the mixture's use and prevent the beetle's reaction chamber from overheating. This pulsation, typically occurring up to 500 pulses per second, allows the beetle to conserve its chemical reserves and extend the duration of its defense capability. This spray is not merely a physical deterrent, but a chemical one as well. The ejected fluid contains concentrated levels of benzoquinone, known for its noxious properties, which serves as a strong irritant to predators. This chemical not only causes a burning sensation, but can also temporarily impair the predator's vision and respiratory function, providing the beetle with a crucial window to escape. The Bombardier Beetle's ability to synthesize and withstand such an intense chemical reaction within its body is an extraordinary feat of natural engineering. The biochemical pathways involved in synthesizing these compounds, the structural adaptations of the beetle's anatomy that withstand these violent reactions, and the neural controls that regulate the precise timing of the spray are all finely honed through millions of years of evolution. Beyond its immediate defensive function, the study of the Bombardier Beetle's unique chemical weaponry offers broader implications. By examining the robust biochemical mechanisms and the beetle's physiological tolerance to high temperatures and corrosive substances, researchers can uncover new insights into materials, science, biomimetics, and even robotics. This shapeshifter can camouflage into anything on the deep ocean floor. At number 5, deep within the complex tapestry of the ocean's ecosystems, thrives a marvel of evolution, the mimic octopus, Thaumoctopus mimicus. Discovered in the murky waters off the coast of Sulawesi, Indonesia in 1998, this remarkable cephalopod exhibits an unparalleled ability to morph its body and behavior to impersonate other marine species, manipulating both predator and prey interactions to its advantage. The mimic octopus is a relatively small octopus with a total length up to 60 centimeters, but its abilities are monumental. What sets this octopus apart is its dynamic method of camouflage, the ability to mimic more than 15 different marine species, including but not limited to the venomous lionfish, sea snakes, and flatfish. This mimicry is so precise that it not only involves changing color and texture, but also adopting the locomotion and distinctive behavioral traits of potentially threatening marine creatures to avoid predation or to sneak up on unsuspecting prey. The biological underpinnings of this mimicry involve a highly specialized nervous system and muscular structure that allows rapid changes in skin color and texture. The octopus's skin is embedded with thousands of chromatophores, pigment-containing and light-reflecting cells that can change the color of its skin in milliseconds. Beneath the chromatophores are layers of muscles that can alter the texture of its skin to match the appearance of a more textured animal, such as a spiny lionfish or a bumpy sea star. Behaviorally, the mimic octopus takes on the form of a lionfish by splaying out its arms and adopting its undulating swim pattern, making itself appear larger and more dangerous. When mimicking a sea snake, it hides 6 of its arms and moves the remaining 2 in opposite directions, imitating the snake's swimming motion. To impersonate a flatfish, it flattens its entire body, moves with a slow, undulating motion close to the ocean floor, and alters its coloration to match the sandy bottom, effectively disappearing from sight. This species' ability to mimic such a wide array of other animals is not just an automatic response, but a calculated decision. Scientists believe that the mimic octopus assesses its environment and potential threats or targets, and then selects an appropriate guise based on the context. This level of behavioral sophistication suggests a cognitive complexity that is quite advanced for invertebrates. The mimic octopus's habitat also plays a critical role in the necessity and effectiveness of its mimicry. Residing in shallow, sandy, and muddy seabeds of the Indo-Pacific, an area bustling with diverse predators predators and prey. It encounters frequent threats that require a versatile repertoire of defense mechanisms. The mimic octopus utilizes its mimicry not only to hide, but to openly deceive both predators and prey to ensure its survival and reproductive success. The James Bond of the sea comes in at number 4. In the intricate labyrinth of coral reefs and seafloor sediment, a small but formidable creature wields a power that defies the ordinary constraints of the marine world. Known as the pistol shrimp, or by its scientific family name Alpheidae, this tiny crustacean possesses a weapon of extraordinary acoustic force. The shrimp's snapping claw, a marvel of natural engineering, can be closed with such velocity that it generates a cavitation bubble, culminating in sonic shock waves that can stun larger prey and even shatter glass. The pistol shrimp, typically measuring only 1 to 2 inches in length, carries one oversized claw constituting up to half of its body weight, which functions much like a biological sonic cannon. Unlike typical claws, this specialized appendage does not pinch, but rather snaps shut at speeds that can reach up to 100 kilometers per hour. This rapid snapping motion creates a high-pressure cavitation bubble as water rushes to fill the void left by the claw's swift movement. The collapse of this bubble releases a formidable burst of energy in the form of a sound wave that can reach up to 218 decibels, briefly rivaling the loudness of a gunshot. This sonic explosion is not merely loud, but also momentarily generates temperatures of over 4,700 degrees Celsius, 8,500 degrees Fahrenheit within the bubble, nearly as hot as the surface of the sun. This phenomenon, known as sonoluminescence, occurs because the bubble's implosion increases the pressure and temperature of the vapor within to extreme levels, emitting a flash of light that lasts a mere fraction of a second. The primary function of this explosive capability is predation. The shock wave produced is powerful enough to stun or kill small fish, allowing the shrimp to capture them easily. This is a critical adaptation for survival in the competitive and predator-filled coral reef environments where these shrimp dwell. Additionally, the pistol shrimp uses its sonic weapon defensively to deter predators and competitively to assert dominance within its territory. The mechanism behind this superpower involves not only the shrimp's physical anatomy, but also its precise timing. The shrimp cocks its claw in a way similar to a tension spring. When released, the claw snaps shut, and the specialized plunger-like structure on the claw socket drives the water out, creating the cavitation bubble. The accuracy and rapidity of this action require fine muscular control and neural coordination, highlighting the shrimp's evolutionary refinement. The effects of the pistol shrimp snap can be disruptive not only to its immediate vicinity, but also to human technology. In naval history, schools of snapping shrimp have been known to interfere with sonar operation, their collective snaps creating a cacophony of underwater noise that can mask the sounds of submarines and other vessels. Ecologically, the pistol shrimp is a keystone species in the reef environments. Beyond hunting, the shrimp's burrowing activity, which it undertakes to create a habitat, helps aerate the seafloor sediment, promoting nutrient cycling and supporting the complex ecosystem dynamics of the reef. At number 3, we have the wolverine of the rainforest. In the dense and humid rainforests of Central Africa, there lurks a creature whose defensive abilities seem almost supernatural. The hairy frog, also colloquially known as the horror frog or wolverine frog, Trichobatrachus robustus, has a gruesome yet fascinating method of self-defense that involves breaking its own bones to produce claws that can pierce through its toe pads. This remarkable adaptation not only underscores the extremes of evolutionary innovation, but also imbues the hairy frog with a capability reminiscent of comic book superheroes. The hairy frog is a relatively large species, growing up to about 11 centimeters in length. Its common name derives from the hair-like structures that appear along the body and thighs of breeding males. These filaments are actually dermal papillae that increase the surface area of the skin, likely helping in the absorption of oxygen through the skin during the mating season. However, it is the frog's extraordinary defensive mechanism that truly sets it apart. When threatened, the hairy frog contracts a specific muscle in its hind legs, which is connected to a small bone in the toe. Normally, this bone is integrated into the toe structure and assists in locomotion. However, under duress, the frog's muscle contractions are powerful enough to break this bone, causing it to protrude through the skin as a sharp, claw-like structure. Unlike most animals that produce keratin-based nails or claws, the hairy frog's claws are actually bone, sharpened by the force of the muscle contractions and the breakage of the bone itself. This self-mutilating defense strategy is as effective as it is horrifying. The claws, once protruded, can be used to lash out at attackers, providing a means of deterrence and escape. After the threat has passed, the frog's body can heal over time. The broken bone retracts back into the toe pad, and the tissue around it heals, although the specific details of this recovery process remain not thoroughly documented, suggesting an area ripe for further research. The mechanism by which the hairy frog breaks its own bones to form claws is an extreme example of what biologists call an evolutionary trade-off. The ability to suddenly produce a weaponized body part offers a significant survival advantage, likely compensating for the frog's relatively exposed and vulnerable terrestrial lifestyle. Predators that might otherwise consider the frog an easy meal are met with a surprise defensive weapon that could cause significant injury or deterrence. The presence of such a mechanism raises intriguing questions about the pressures faced by this species in its native habitat. Living in a region populated by numerous predators, the evolution of such a defense suggests a high predation risk that favors individuals capable of such extraordinary self-defense. This adaptive response highlights the complexities of evolutionary biology and the often drastic paths that organisms can take to ensure survival, just like Piccolo out of DBZ at number 2. Deep within the mystical waters of Xochimilco, a network of ancient canals near Mexico City, dwells a creature with capabilities so profound that they seem to defy the very laws of biology. The axolotl, Ambystoma mexicanum, a species of salamander, possesses regenerative abilities that border on the miraculous. Unlike any typical creature, the axolotl can completely regenerate lost limbs, tails, and even more complex tissues, such as parts of its heart and brain, all without any scarring. This extraordinary capability makes the axolotl not just a subject of fascination, but also a significant focus of scientific research. The axolotl's regenerative process involves dedifferentiation, a phenomenon where adult cells revert to a stem cell-like state. These stem cells, known as neoblasts, can then multiply and develop into different types of cells as needed, allowing the axolotl to replace damaged or lost tissues with remarkable precision and efficiency. The scope of the axolotl's regenerative powers is vast. It can regenerate not only limbs and tail, which include complex structures of bone, muscle, nerve, and vascular tissues, but also parts of internal organs that are often considered irreparable in most other vertebrates. For instance, the axolotl can recover from a damaged heart, restoring its function completely through the regrowth of cardiac muscle and other heart tissues. Similarly, injuries to the spinal cord, which would typically result in permanent disability in other animals, are fully repairable in the axolotl. This salamander can even regrow parts of its brain, an ability that remains one of the most astonishing feats of regeneration observed in the animal kingdom. From a scientific perspective, the axolotl's regenerative process is regulated by a combination of growth factors and hormones that signal the cells at the site of injury to commence the healing and regrowth processes. Researchers have identified several key proteins and genes involved in this signaling process, such as the Lin28 gene, which plays a crucial role in maintaining the proliferation of stem cells needed for tissue regeneration. Mechanisms underlying this regeneration are not just rapid, but also incredibly precise. The regrowth of a limb, for example, follows the original blueprint of the limb's structure, ensuring that each bone, muscle, and nerve is recreated accurately. This process can take weeks to months, depending on the extent of the damage and the environmental conditions, but the result is a fully functional appendage, virtually indistinguishable from its original. The axolotl's ability to regenerate is so refined that it even surpasses those of other salamanders, which themselves are known for significant regenerative capabilities. Scientists believe that the axolotl's retention of its larval features into adulthood, a trait known as neoteny, may contribute to its enhanced regenerative abilities. The axolotl rarely undergoes the metamorphosis that other amphibians do. Instead, it retains gills and remains aquatic throughout its life, which may create the stable internal conditions favorable responsible for complex tissue regeneration. Beyond its biological marvels, the axolotl holds a revered place in mythology and culture, often associated with the ancient Aztec god Xolotl, who was said to have transformed into this amphibian to escape his enemies. While the legends surrounding the axolotl amplify its mystique, the real-life capabilities of this salamander challenge our understanding of biological limits and offer promising insights that could one day lead to revolutionary advances in medicine, particularly in the fields of organ regeneration and wound healing. At number 1, we have the tiny alchemist of the rainforest. In the dense rainforests of South America, a marvel of natural engineering and agricultural prowess thrives within the undergrowth. The leafcutter ant, genus Atta, commands a complex society that engages in one of the most sophisticated forms of agriculture practiced by any non-human species. These ants cultivate fungus gardens, meticulously maintained and fed with freshly cut plant material, which in turn nourishes an entire colony that can number in the millions. The leafcutter ant's agricultural system is a stunning demonstration of evolutionary innovation and communal living. These ants cut leaves not for consumption, but as a substrate to grow specialized fungus, which they cannot live without. The ants have a symbiotic relationship with this fungus, which is so integral to their survival that each new colony begins with a fragment of fungus carried by a female ant from her birth colony when she flies away to start her own. Each leafcutter colony functions like a superorganism, perfectly coordinated through a division of labor that is finely tuned to the ant's size and age. The larger, more powerful ants venture out of the nest in impressive foraging lines that can stretch up to 100 meters, cutting pieces of leaves and flowers, which are notoriously difficult to break down chemically. These foragers are not merely collectors, but also highly selective botanists, choosing only certain types of leaves that are known to foster the growth of their fungal crops. Once the leaf fragments are brought back to the nest, smaller worker ants take over, further cutting the leaves into smaller pieces with their sharp mandibles. Subsequently, even smaller ants chew these fragments into a pulp, which serves as the ideal substrate for the fungal growth. This processing of plant material into a form suitable for fungal cultivation showcases the ants' intricate knowledge of organic farming practices. The cultivation process is equally fascinating. The ants actively manage their fungal gardens, adding leaf pulp to grow the fungus, while also secreting antibiotics from their bodies to control other fungal species that might compete with their crop. Moreover, they use their own fecal matter, which contains digestive enzymes, as a fertilizer to promote fungal growth, demonstrating an understanding of biochemistry that rivals human agricultural techniques. The fungus, in turn, produces gongylidea, swollen hyphal tips, which the ants harvest and feed to their larvae and queen. This mutualistic relationship is so advanced that the fungus grown by the ants does not exist anywhere in the wild. It has co-evolved with the ants to such an extent that each is entirely dependent on the other for survival. The architectural mastery of the leafcutter ants extends to the construction of their nests, which are underground marvels that can extend up to 30 meters in depth and house millions of individuals in chambers connected by complex tunnel systems. These nests maintain a precise microclimate essential for fungal growth, with ventilation systems that regulate temperature and humidity. Such sophisticated agricultural practices not only underline the leafcutter ants' ecological role, but also highlight their impact on forest dynamics. They are key agents in nutrient cycling and soil aeration, enhancing the ecological health of their environments.